Hello students, in this lecture we will see some of the properties of the cell membranes, what is their structure, what's the function, and also some of the history of the cell membrane. So the cell membranes, basically, if you want to define it, so it is a kind of boundary for the cell, right? And it separates the cell from its surroundings. That's the main concept. And they're always selectively permeable. What does that mean? That only selected material will be able to cross the cell membrane, and only the selected material will be able to get out of the cell, right? Both ways. So both ways, the transport of the molecules in and out of the cell will be will be performed by the cell membrane. Why cell membrane is selectively permeable? We already have discussed that if the cell allows everything to go inside it, then there will be a mess within the cell, right? And if the cell is unable to contain the organelles, and if the cell is unable to restrict the movement of the cell constituents, then what will happen? That all the cell constituents will leave the cell, and again it will be a disaster for the cell. So that's why the cell membrane, it is selectively permeable, that in and out transport of the components, it is highly specialized, and it is highly selective. So here is the structure. We already have discussed about the structures in the previous lectures, but here this is amphi. It is composed of lipid bilayer and amphipathic molecules. Amphipathic molecules means that they may have partially positively charged, they may have a partial negative charge, and they may be hydrophilic, they may be hydrophobic at the same time, right? So the inner layer of the cell membrane, inner layer means the membrane, the layer that faces the cytoplasm, and then we have an outer layer of the cell membrane that faces the extracellular matrix. Both these layers, they are hydrophilic because the outside environment is aqueous, the inside environment is aqueous. Because of this aqueous environment, these two portions of the cell membrane, they are they are polar and they are water loving, right? So which part of the cell membrane is water hating? And that part is here. You see these lines, these layers, these are the balls, these are the polar heads, and these are the tails. These tails actually are hydrophobic, right? And hydrophobic and nature of these tails will restrict the movement of molecules in and out of the cell membrane. This is a great arrangement by the nature. Okay, if this membrane, if this inner part has been hydrophilic, then everything will go in and out of the cell, right? So here you see it is restricted because of the hydrophobic nature of these tails. So it is a phospholipid bilayer, and phospholipid molecule, as you all know, that it consists of two long non-polar tails. As you can see here in the in this image, these are the tails, these are non-polars, and these are actually hydrocarbons, and these are lipids. And hydrocarbons, they, because of their structure, they play a part in the hydrophobicity of this molecule, right? And the head, as you can see, the orange head, it has phosphate, right? And because of this phosphate groups, its nature is water loving, right? Because of this presence of this phosphate groups, they are hydrophilic in nature. So this is the combination of positively charged as well as negatively charged, hydrophilic as well as hydrophobic portion that gives cell membrane its unique structure. So lipid bilayer, it has lots of things embedded in the layer, right? And here you see the electron micrograph of the membranes of the two adjacent cells, that these membranes, they have communications with each other as well, right? And here you see these hydrophilic channels, these are hydrophobic area, so the hydrophilic things will not be able to cross this barrier. So for that, nature has built tunnels. These tunnels are hydrophilic in nature. Okay, so hydrophilic stuff, hydrophilic molecules, if they want to get in or out of the cell, they will be using this tunnel. And of course, this tunnel is not an open gate. There is a criteria that at what time these tunnels will be open and at what time these tunnels will be closed, and their opening and closing times, they are directly related with the physiology of the cell, the need of the cell, and there will be some kind of security arrangements so that these gates should not be open forever or closed forever. So on and off, these gates will provide these gates built on the tunnels, they will provide the transport of the things needed in and out of the cells, right? So here you see the green carbohydrate side chain. These green carbohydrate side chains, these are these are responsible for the identification of the cells, right? And hydrophilic channels, hydrophilic regions, along with the hydrophobic regions, it constitutes the lipid bilayer of the cell membrane. These are the functions 1, 2, 3, 4, 5. These 5 functions, they are illustrated here very briefly. That the cell membrane, it serves a boundary of the cell, and it helps to organize. If there is no membrane, then there will be no organization of the cells. The cell has lots of organelles in its in the cytoplasm, right? And then the transport of the things in and out of the cell, it is also monitored by the cell membrane. Then the signal detection, cell has molecules that can detect external signal, for example signal from the brain or signal from the hormone, that what is required by the cell. Okay, and then these cells will respond to these signals, and they will give a response. For example, if some hormone is going to bind it, the cell is going to give response. If that hormone is for the division of the cells, the cell will divide. If that hormone is for the growth of the cell will grow, right? And here the last function is the cell to cell interactions that are also very important in the signaling. Phospholipids, they are, as we already discussed, they are the most abundant lipid in the plasma membranes. They are the lipid molecules and they having phosphate group attached to them. These are having hydrophilic as well as hydrophobic nature at the same time. Fluid mosaic model. But what does mean? What does mosaic mean actually? Mosaic, I will give you a very simple example of the mosaic. For example, you have a ball, a plastic ball, and you put some kind of needles in that ball, right? And if those needles can move around here and there on the surface of the ball, that will be a mosaic, right? And it is a kind of model where the cell membrane, it can perform lots of functions because of the movement of those embedded molecules. Those embedded molecules, they are not fixed on the surface of the cell membrane. They can move like a fluid. Okay, so this fluid mosaic This slide is showing the history about the cell membrane. There people used to study the cells, and here are some of those names of the scientists who perform these activities, and they were keen to know how cell membrane is working, right? So after the discovery of the cells back in the 17th to 16th century, so people were looking into the cell membrane. So first of all, the Overton, he said that it is composed of a lipid, and then the next scientist, they said no, it is yes, it is composed of lipid, but it is just a single layer. Then the coming scientists, they said no, this is a lipid bilayer actually, and so on. So lipid bilayer. After that, this era, 1940s, this is the era where electron microscope was there in the 50s and 60s, and there was a lot of use of electron microscopy in all the fields of the sciences like physics, chemistry, and the biology. So here the electron micrograph here shows that it is a kind of unit. So they call it as a unit membrane, and as time progresses, then we have Singer and Nicholson. They said that it is a fluid mosaic model. Mosaic means, as I already told you, that stuff are embedded in any material. Okay, so these cells, they are not liquid cells, these are solid cells, right? And fluid means these solid cells can behave like fluid. This is what it is all about, the fluid mosaic model. Then in the 1980s, the membrane protein structures, they were discovered that there are some tunnels, and the hydrophilic channels, they were discovered that even if it is lipid bilayer, but still the hydrophilic things can move in and out of the cell without any problem because of the presence of these hydrophilic channels, right? And then currently lipid raft means a particular area having a particular thing. So here raft means that in particular areas of the cell membrane there are a particular molecules present, and those molecules are responsible for a particular function. For example, the cholesterol molecule, it was discovered later on that the cholesterol molecules, they perform a fluidic function, and they are responsible for some of the fluidity of the membrane. So this slide is showing the inner structure of the cell membrane. Cell membrane, it is basically an envelope. Right here you see a sandwich-like structure. We people eat sandwich, and when we have two slices, and between those slices we have mayonnaise or egg or maybe shami, right? And when you just remove the top layer, you will see the inner structure. This inner structure here, it is called as E phase. E phase means this is the outer membrane, and this layer is facing the outer structure of the cell, the cell that is facing towards the extracellular matrix, right? And this structure, this one, you see the mouse here, the arrow, this is called as the P phase. This is the inner monolayer of the cell membrane. So these are two different methods to describe the different surfaces, and these surfaces, they are involved in the transport of different materials in and out of the cells. And here you see more detailed shape of this lipid bilayer. Here you see these are embedded glycoproteins, and these are the hydrophilic channels here, and these carbohydrate side chains responsible for the identification of the cells. Then there are some proteins who are anchored, lipid anchored membrane proteins, and they have different functions. Sometimes they leave this anchorage. At most of the time these proteins, they're just anchored over there to perform different signaling functions. And in the chapter 14 we will see that how these proteins that are embedded, how they perform functions, and most of the proteins that are embedded, they are actually the receptors. Okay, receptors means they are going to receive something from the outside world. Okay G, now we move towards our next slide, and next slide is showing about the fluidity of the membranes. As I told you that these are actually the cells are solid cells, but they behave like liquids, and the movement is going on even in the phospholipids. Here you see this movement is called as the lateral movement occurs. So lateral movements, this molecule can go here and this molecule can go here, and here this is a flip-flop movement of the lipid molecules. So this molecule can go here and this molecule can come here. So sometimes they can cause some mutation as well. There are some rare diseases that can be originated because of the abnormal movements. Okay, so this flip-flop movement is rare, but this is this one is, this lateral movements, which is a huge number. They have 10 raised to the power 7 times per second. Okay, so this movement actually, what is the significance of this movement? This movement gives the cell its fluidity, right? Fluidity, where when they keep moving and they keep moving, and that movement helps in the transport of the stuffs in and out of the cell membrane. Okay, integral membrane proteins, then we have peripheral membrane proteins, then we have lipid anchored membrane proteins, and as I told you, and what is the difference between integral membrane and the transmembrane? Transmembrane is a kind of membrane that can cross both layers of the cell membrane, right? We have two layers, as I told you, E phase and P phase. It can cross all the surface of the lipid bilayer. So most of the receptors, they are transmembrane receptors, like the G protein coupled receptors, because they have one projection outside the cell, the other projection is inside the cell. So these proteins perform various functions. And then we have some lipid anchored membrane proteins. They are present on the E side as well, they are present on the P side as well. Okay, so they perform different signaling functions, and and those signaling functions actually are responsible for the physiology of the cell, for the proper functioning of the cell, or for the proper work that is needed by the organism. It is performed needed by the organism. The organism wants its cells to perform according to the need. When we are hungry, the cells will perform differently. When we have eaten the meal, cells will perform differently. And when we are doing some exercise, then the cells will perform differently. So these are different physiology. And when we are ill, we have a pathological condition, then the cells will perform differently. So these molecules, they will behave, they will perform differently in different situations. Okay, this slide we already have discussed that the proteins, that determines the functions, specific functions in specific conditions, right? And membrane proteins and their functions, their peripheral proteins, integral proteins. With this one we already have discussed in the previous slide, where I have shown you the transmembrane proteins and the integral proteins and the anchored proteins. These are the summary of some of the functions, right? Major functions: transport, enzymatic activity, signal transduction, cell to cell recognition, and the attachment to the cytoskeleton, intercellular joining, lots of extracellular matter. This is a summary of the functions of the cell membrane. Okay, and in the coming slide I will show you that what types of functions they are performing with the help of image. So here is the image, like for the transport, it is, and sometimes the energy is needed for the things that will go inside the cell, and there are some these blue balls, they want to go out, right? And sometimes this movement may need energy, and sometimes it will not need energy. So concentration gradient, if it is based on the concentration gradient, then it will not need energy, like the movement of molecules from the higher concentration to the lower concentration, that that will not need energy. Energy will be needed when there is an uphill movement. Okay, the area of the movement of the molecules from the area of lower concentration to higher concentration, that will need ATPs, right? So here you see the transport, again the cell membrane is involved. This is again a single cell, and enzymes are there. The enzymes, different enzymes, they are going to bind on the surface of the cells, on the cell membrane, and they will cause the change in the physiology of the cell. Whatever message that hormone is bringing from the central nervous system or from the different glands, they are going to ask the cell to perform that specific function. So signal transduction means that the signal is transferred from one location to another location, and how the signal is processed, how that signal is received. The signal is received by a receptor molecule that is present on the surface of the cell. Okay, and this is a signaling molecule. This signaling molecule, it is called as ligand. This ligand is going to bind with this receptor, and then this receptor will go through different conformational changes. The signal is then transferred to the cell to perform specific functions. Okay, and then cell to cell recognition. I told you that the glycoproteins, whenever there are carbohydrates attached to the proteins that are embedded on the cell, so these carbohydrate molecules, they always perform the function of recognition. For example, here this may be an immune cell, and this one glycoprotein, it is present on a non-immune cell. So immune cells, they will have some mechanism by which they can recognize these cells, that these cells belong to the organism and these are not outsider. If there are no carbohydrate chains on the cell, then this cell is engulfed by the immune cell. Okay, and then intercellular joining. Sometimes cells need to make tissues. Okay, and they have to perform functions together, so they will perform the same functions as a group. So intercellular joining will be important in that part. So attachment to the cytoskeleton and extracellular matrix. The cell membrane also perform these functions. Extracellular matrix, I have told you before that this is also a kind of liquid that is responsible for the proper performance of that particular cell. Okay, so the molecules on the cell membrane, they have linkages with the extracellular matrix as well. They will receive the signal from the extracellular matrix, as well as they can send their own signal to the extracellular matrix. So how do we analyze the membrane proteins? That's very important. Okay, this might be included in our lab as well. The SDS page, sodium dodecyl sulfate polyacrylamide gel electrophoresis. It is abbreviated as SDS page. It is a technique used for the analysis of the proteins. Okay, almost every cell biology lab has this apparatus, SDS page apparatus, and they perform it in the lab to know that which protein is there, what is the size of the protein. Here we can also measure the size of the protein, right? And based on the size of the protein, we can go for further analysis that what could be this protein, and then we have some staining, that how many proteins we have. Here we have a marker, the multicolor bands. These are indicating different molecular weight molecules of the proteins. Okay, so here we will see that if there are protein molecules, they have traveled some distance in the gel, and we can measure the size of these molecules by comparing it with the already known sized molecules that have traveled from the beginning of the gel to the end of the gel here. Okay, so most of the time the molecules that travel from the negative to positively charged ends. So we will load the samples here, and the samples, then we will put the electricity here, the power supply from negative to positive charge. The molecules will move from the negative to positive charge, and they will be resolved, and they will be analyzed according to their molecular weights. This is one technique that we use for the analysis of the protein. Okay, it is very famous. And then another technique that is known as X-ray crystallography. This is very advanced technique, and it is also very difficult. So what we do here, we use X-ray beam, and that X-ray beam is bombarded on the crystallized molecule. To make this crystal is very difficult. It is very, very difficult to make a crystallized molecule. So once you have the crystal, then this crystal can give different projections, different diffracted response on the X-ray plate. So based on this data, the scientists, they are going to have a prediction that what could be the pattern of this crystal. Okay, so this crystal mostly these crystals are studied like that, and using this information we can also know the shape of the molecule, the structure of the protein molecule, right? STS page tells us the size of the protein molecules, and here X-ray crystallography, that it can show us what actually is the shape and structure of the protein molecule, right? So these two techniques, they are mostly used in the cell biological experiments and the cell biology labs. Okay, so if you have any questions, please send an email. Also you can drop me a message on Skype as well, that we have we have a group, Skype group as well. So this was all about the chapter 7, I think, and hope you guys are understanding it, and still if you have any problem, please do contact. Okay, thank you very much, see you later.